Hey everyone, it's Mr. Veve, and this lesson is on genetic engineering. So let's get right into it with our first key concept. Scientists study the genomes of organisms in order to discover what genes can be changed and what the outcomes of those changes could be. So we're going to look at a few different examples of how humans have been able to modify the genome or certain genes of different organisms and the different ways in which we do that. So let's start with the first one, which is selective breeding. Now, this is a deliberate attempt by humans to pass desired traits on to the next generation. Now, one of the ways that we see this is in dog breeding. Now, I have an example here of a pug, what it looks like today on the left-hand side, and what it looked like in 1880 on the right-hand side. And you notice quite a few differences. Okay, the snout is not quite as prominent as it was in 1880. It's a little bit more smushed, and the face is darker black, and the coloring is a little bit off. But they kind of look like the same dog. But what's happened is over years and years and years and generations, certain traits were considered desirable for a pug to have. So what humans would do is they would take pugs that have those certain traits, and they would make sure they breed together to produce offspring that had those traits more closely resembling what they wanted. So that goes on for years and years and generations, and you end up getting an organism or a breed of dog that does not really resemble what it originally looked like over 100 years ago. It's fun to look some of these up, and you can see especially English bulldogs then versus now. So it'd be a fun thing to look up. Next thing we're going to talk about is hybridization. That's when you cross two different species that share common genes. These hybrids, we have to say, are usually sterile, meaning they cannot have children once they are created. They can't have offspring. Here's a few examples. A Zedonk is a zebra with a donkey, so you can see that one on the left side. A camel and a llama is a comma, and a lion and a tiger is a liger. Now, with the liger, that is a male lion with a female tiger. If you switch that up and you have a female lion with a male tiger, you'd actually call it a tigon. It does make a difference what you call it as to which organism has the male or the female. But just a few different hybrids that humans have created here by mating two different species that do have a lot of common genes. So next, let's talk about just genetic engineering in general. So if we are able to isolate genes, one thing we can do is we can transform an organism, or we could even clone an organism. So let's look at cloning first. Now, the thing on the left here is the famous experiment with the sheep named Dolly. Now, this is something that we actually have accomplished as humans. What scientists did is they took cells from the sheep, the white-faced sheep on the left, and they took an unfertilized egg from the sheep on the right, who has a black face there. And they took all of the DNA of that sheep out of the unfertilized egg, and they fused the two cells. So the DNA from the white-faced sheep went into the unfertilized egg that had no DNA in it anymore. So it effectively replaced the DNA, and that embryo was then implanted back into the surrogate from which the egg came. And what happened is you got an exact clone of the sheep from whose cells you took previously, and they named that sheep Dolly. So they show how it would happen in humans on the right-hand side. We haven't done this yet, where you just completely clone a human being. So one thing I want to point out is there are some studies that suggest that cloned animals may suffer from certain defects and maybe health problems that we don't really know. Cloning humans would actually also raise some serious ethical and moral issues. So those are some things to think about on your own. How do you feel about the ethical and moral issues that would come about if someone were to clone you or clone a loved one? So really, really something that you have to think about in the world of science here now that we're getting closer to this kind of technology. In fact, it's even happened already. So let's talk about transformation. So we talked about cloning, and now if we can isolate genes, we can also transform cells. This is actually very, very helpful. So a cell transformation is when you take cell from outside, you take a DNA from outside the cell and you implant it into the DNA of something. So in this case here, we're going to look at a bacterium, and they have a circular piece of DNA called a plasmid. Now what you can do is you can actually cut this circle open and insert a new gene. So that's what we're doing here. You cut open that red plasmid and insert that green DNA, and now you have this new DNA within the bacterium. So whenever the bacterium replicates itself, it's going to make that new plasmid, that new DNA. So this organism, when it has new DNA in it, is called transgenic, and that new DNA is called recombinant DNA, so recombined DNA. So let's look at an example of how that actually works for us. So one thing we use this for is actually producing insulin for people that need it, so type 1 and type 2 diabetic patients. So you have a bacterium that has that circular DNA. What scientists will do is they cut open that plasmid and they insert the gene that codes for insulin production. So they insert that in there, and so when the bacterium starts dividing, it divides into more bacteria that can actually make insulin. And this happens millions of times, and what you have is just a farm of bacteria that produce insulin that can be refined and then given to patients that need it. So next thing we're going to talk about is genetically modified organisms, and in this case, genetically modified plants. These are all over the place. There's ways that we can modify certain plants to make sure that they can produce a natural insecticide. This keeps them safe from being eaten by insects. Also helps maybe help them produce larger yields of crops per year. Now, these are all by coding for and making sure certain genes make it to the next generation. Another way is looking at polyploidy. That means having more than one set of homologous chromosomes. In this case, in the strawberry, a normal strawberry fruit is very small, but if you genetically modify it to have two sets of homologous chromosomes, so it has 4 copies of each chromosome instead of just 2, you can actually make the fruit quite a bit larger. So again, this comes down to how do you feel about eating plants that have been genetically modified? Do you think there's any moral or ethical issue with that? So something to think about. So finally, we look at genetic screening and genetic therapy. So genetic screening really is just a way to detect genetic disorders by testing the DNA. And this can be done for a number of things, like seeing your risk for certain types of cancer or other types of inherited disorders. And this can lead to potentially therapy, gene therapy, where you replace missing or defective genes with new ones that are healthy. This is still somewhat controversial. We don't know how far we should go with cutting up people's genes and inserting new ones, because that could lead to some, again, moral and ethical issues.